Today we have a special guest with us, Dr. Jessie Miller. Dr. Miller got her PhD in nutritional sciences and also my alma mater, so always glad to see the nutritional science people here. And right now she is here at the U of A as an R25 Cancer Prevention and Control Training Fellow. And so as part of what they need to do as part of our program is to present their work to everyone at Cancer Prevention and Control and the Cancer Center. And that is what Dr. Miller will be doing today. She's talking about metabolomics for breast cancer prevention. And metabolomics, for those of you who may not be in the nutrition cancer area, it's becoming a very hot topic. And it's also very complicated. So this should be a very interesting talk. So thank you, Dr. Miller.
Thank you, Beth. Yeah, hopefully I don't confuse everyone with my slides, but I have a lot of data to show you, and I think it's exciting. So I'm going to talk about metabolomics and how we've been trying to utilize this new technology to improve our strategy for breast cancer prevention. And so for those of you who don't know, I thought I should define metabolomics. And so metabolomics is the measurement of metabolite concentrations and fluxes and secretions in the cells and tissues in which there's a direct connection between gene expression, protein activity, and metabolic activity itself. So basically, metabolomics is the study of how all these metabolites are interacting with a living organism. So your metabolome is just the profile of those metabolites. And then the description of that profile would be your metabotype. And so there's always a lot of new words that come out anytime there's a new technology. So your metabotype is your metabolic phenotype. And so we can kind of think of, well, we're trying to discover what is a cancer risk metabotype. And so you can think of, well, maybe inflammatory metabolites would indicate a cancer risk metabotype. And so metabolomics is already kind of used in clinical practice to characterize metabolic disorders with newborn screening. At the U of A, we screen for around 20, but one of my friends and colleagues at Imperial College has developed an assay to screen for over 200 at one time, and she's trying to move that forward in clinical practice. Another interesting application of metabolomics is to understand the human relationship to the microbiome. And so we have 10 times as many bacteria in our gut and on our skin as we do our own cells. And so these bacteria are metabolizing food we eat. They have a pretty profound effect on our gut and our overall health. And so it's important to understand how their metabolites are interacting with our own health. And so what I'm going to show you today is how we can use metabolomics to assess response to an intervention. And we can look at toxicity, efficacy, as well as mechanisms in one assay. And that intervention can be diet or chemotherapy. And in this case, I'll talk about chemoprevention in a clinical trial where we gave early-stage breast cancer patients limonene. And I'm also interested in new biomarkers and nipple aspirate fluid. So halfway through the talk, I'll kind of switch gears and show you some of our preliminary data with NAF. So limonene is a terpene, and it's found in the peels of citrus. And my whole graduate career was with limonene, and even after all this time, I still can't really pronounce the chemical name, but there it is for you. It's found in citrus peel. It's very lipophilic. And because of its lipophilicity, we're interested in its anti-cancer activity for the breast. It's also demonstrated anti-cancer activity in skin, gastric, lung, and oral cancers, as well as leukemia and lymphoma. So like every other bioactive food component or dietary intervention, limonene has a laundry list of mechanisms. This include P450 modulation, farnesyl transferase inhibition. The most convincing preclinical data is inhibition of the TGF-beta1 mannose-6-phosphate IGF-2 receptor. It also inhibits PGE2. It may have some antioxidant activity, has lipolytic effects. It's been proposed to be an immune modulator, and it induces apoptosis. And so I think it's important to relate these mechanisms back to the hallmarks of cancer and how they, and so I think most people are familiar with this diagram from the Hanahan and Weinberg paper in Cell in 2011. And so we have the hallmarks of cancer: sustaining proliferative signaling, avoiding immune destruction, resisting its cell death, tumor promoting inflammation, et cetera. So the ones that limonene may affect are, so may inhibit sustaining proliferative signaling through its farnesyltransferase inhibition, resisting cell death by inducing apoptosis, inhibiting tumor promoting inflammation through PGE2 inhibition. Because it has antioxidant activity, it may prevent genome instability, and then avoiding immune destruction through its immune modulatory activities. And so these are all proposed mechanisms, and I would say the preclinical evidence for each is, not super strong. But I think, so metabolism as a whole is really related to also the hallmarks of cancer, and I think we're all by now familiar with the Warburg hypothesis, where a cancer cell will uptake glucose at a very high rate and produce a lot of lactate. And then that lactate has an effect on avoiding immune destruction, evading growth suppressors, and invasion and metastasis. But also with this enhanced proliferative signaling of cancer cells, you have induced PI3 kinase, AKT, and mTOR, and then you have a change in the profile of amino acids, and actually in a lot of metabolomics studies, the profile of amino acids has indicated, has differentiated between cancer or not. Along those lines, kinurins are tryptophan breakdown products that help a cancer cell avoid immune destruction, and then there are other pathways. But there are also metabolites that are systemic that may affect the cancer itself. As you think about a small little tumor, and while it has all these metabolic effects, we may not be able to see them systemically. And so it may be more prudent to look at metabolites that are actually affecting your systemic state, like an inflammatory state, rather than trying to pick up these tiny changes in a tumor in the plasma. And so some of those, circulating PGE2, I like the arachidonic acid pathway, so I also picked HEATs, may promote inflammation. HEATs also can prevent proliferative signaling, and then bioactive food components and their metabolites can also have an effect on this entire pathway. And so I think, and these are things that we can measure systemically that may have an effect on cancer growth. And so I'll take us to the limonene trial that I talked about earlier, and this is the trial where we tried to apply metabolomics. But first, so we have 40 early-stage breast cancer patients electing to undergo excision surgery. At baseline, we collected plasma, and in the plasma measured limonene and ferulic acid, which is limonene's major circulating metabolite, and we looked at IGF axis and adipokine proteins. We collected diagnostic tissue blocks and looked at proliferation and apoptosis markers. Participants were on limonene for 2 to 6 weeks, and the reason for this range in time is because these are early-stage breast cancer patients electing to undergo surgery, and so we couldn't ethically extend the time till their surgery, and so we kept them on limonene as long as they were willing. The average time on agent was 21 days. So participants took 2 grams of limonene daily, and then after the intervention, again, we collected plasma and measured limonene and ferulic acid and these IGF axis and adipokine markers. They had surgical excision of the tumor, and we collected fresh breast tissue and tissue blocks. And then we were able to measure limonene and ferulic acid in the breast tissue to make sure that our agent was reaching the site of interest. And then in the tissue blocks, we look at proliferation and apoptosis markers. In the surgical excision block, we looked at proliferation into apoptosis markers. And so what we found, so over here,
so this is plasma concentration in micromolar. This is breast tissue concentration. You see the scale is different. This is 0 to 12 micromolar. This is 0 to 100. And limonene is in red, pearlic acid in yellow. And you can see limonene is depositing in the breast tissue at levels high that in preclinical models elicited an anti-cancer effect. So our drug is reaching its target at a level that could potentially have an anti-cancer effect. Pearlic acid is not depositing in the breast tissue at all. And in plasma, pearlic acid is higher, which makes sense. It's more hydrophilic. I should note that these samples were taken the next day after their last limonene dose. So pearlic acid, 3 hours after you take limonene, is metabolized very quickly to pearlic acid. So 3 hours would be the peak plasma concentration. But the main endpoint was limonene concentration in the breast, and I think we showed that. We also looked at tissue biomarker expression in the biopsy at baseline and in the surgical specimen. So cyclin D1 is a cell cycle regulator. It kind of pushes cells through the G1 phase, and it's upregulated in breast cancer. And we showed a significant decrease in cyclin D1 from pre to post intervention, and pretty strong p-value. Ki67 is a marker of proliferation, and it did not change at all. And cleave caspase 3 is a marker of apoptosis, and it did not change at all. And so for my R25 project, I wanted to profile plasma pre and post intervention, and then quantify those metabolites, which has yet to be done, and then correlate the magnitude of change in these metabolites from pre to post intervention, and then relate them to the changes we saw in tissue markers. So just to remind you, so we have plasma from pre and post intervention. And so when last I was up here, I gave a very detailed description of methods, and I thought, so this time I would spare you that pain. And just to say that we measured these metabolites using chromatographic methods, and this is an example of a chromatogram. Down here, this is time, and this is abundance. And each peak represents a separate metabolite. The red is pre-intervention. This is one participant. And the purple, which is, the color is a little bit difficult to see the difference, I'm sorry. The purple is post-intervention. So we primarily saw changes in bile acids, lipids, and long-chain lipids. And this is where we left last time I was up here. And so we've gone on and we've identified more specifically those metabolites. And so those classes of metabolites which changed with the limonene intervention were bile acids, sulfate adrenal steroids. We saw changes in energy metabolism. There's an increase in collagen breakdown products. And those metabolites that were significantly correlated with the change in cyclin D1, so the change in the metabolites was correlated with the change in cyclin D1, were carnitines and glycophosphocholines. So now I'll go through each of these pathways, and then and once I get through them, I'll kind of try to bring them all together to make a story. And so we'll start with bile acids, since that's Patty's favorite. And so this pink line is the liver. And we start with cholesterol. Cholesterol is metabolized to cholic acid and chenodeoxycholic acid, which are the major bile acids. And then they're conjugated with glycine and taurine to make secondary bile acids. These are also metabolized by intestinal microbiota. And you get ursodeoxycholic acid, deoxycholic acid, and lithocholic acid. Some of these are excreted, or they enter enterohepatic circulation, come back to the liver, are further conjugated with glycine and taurine, and then all of these circulate. And so here are the circulating bile acids. Again, not all of them, just the major ones. And the arrow indicates this is a significant change overall, and then this in parentheses is the fold change. And so in general, there was this between 1.5 and 3.5 fold increase in circulating bile acids with limonene intervention, which is interesting. The only bile acid that went down is this sulfated one. So cholesterol is also, and then glycine, which is used for conjugation, would also significantly increase 40%. And in nutrition, a p-value like this is like cause for celebration. And so, so this kind of makes sense. It fits together. So cholesterol is also a precursor for adrenal steroids. So we saw a general increase in our bile acids. We saw a slight increase in cortisone, 30%, but this was a non-significant. And so we also saw a decrease in DHEAS, but no change in DHEA. This kind of makes sense that we would see a change in the sulfated form, because the sulfated form circulates at about 300 times higher levels than the non-sulfated. There's also a decrease in prenenyl sulfate. Further down the pathway, we also saw a decrease in 4-indostryrin 3-beta, 17-beta disulfate, and these are all statistically significant. And then a significant decrease in aldosterone sulfate, and a significant decrease in epiandrosterone sulfate. And so this is interesting, and it could either, there are a couple explanations. One, because we saw an increase in bile acids, maybe availability of cholesterol to the adrenal gland is lower, so this is why we see a general slight decrease. Or limonene might just be affecting sulfation because the only adrenal steroids that were decreased were sulfated. So it could be either of those. Or maybe for some reason this increase in cortisone is affecting the rest of the pathway. But anyway, so we also saw a significant increase in collagen breakdown products, which could be good or bad depending on which collagen is breaking down. So collagen is in all of your connective tissue, and it's made up primarily of glycine, proline, and hydroxyproline. Matrix metalloproteinases are upregulated. When matrix metalloproteinases are upregulated, they break down collagen into many peptides. And the primary peptide is prohydroxyproline, and this peptide is pretty specific to collagen. And we saw a 40% increase, and this was statistically significant. These peptides are further broken down by endopeptidases into glycine, again, proline, hydroxyproline, and trans-4-hydroxyproline. Glycine and proline are involved in many other metabolic processes, and like I just showed you. But trans-4-hydroxyproline and pro-hydroxyproline are pretty specific to collagen. And so, again, this is interesting. Many of you are familiar with this pathway. So we have glycolysis, glucose to pyruvate, and then the TCA cycle for energy. And I've only left in the major metabolites to simplify things. We saw a 70% increase in pyruvate, and again, we're measuring all of these metabolites in circulation, but I've indicated where in the cell this activity will happen. We saw a decrease in acetylcarnitine, and this was statistically significant, a decrease in citrate, and an increase in malate. So pyruvate can also be made from amino acids. And so here's glycine again, and alanine was increased. Aspartate, we had a slight decrease. Glucose also has alternative metabolic pathways. And so we saw a significant increase in fructose. And then glucose can also be metabolized to vitamin C. And we saw a 15-fold increase in vitamin C, but there were 2 participants, 2 or 3, that were not fasting, and when we take those out, this increase is still statistically significant, but it's more like a 2-fold increase and not 15-fold. So most likely a couple people just drank some orange juice before they came in. But even when we take them out, that this increase in vitamin C is still twofold and statistically significant, and we see an increase in its precursor, glucuronate, and so that kind of all fits. We also saw a statistically significant increase in beta-hydroxybutyrate, which indicates that lipids are being used for energy rather than glucose. So this kind of makes sense. If lipids are being used, so beta oxidation would supply acetyl-CoA to the TCA cycle, and then glucose would be shuttled off into these other metabolic pathways. So that also sort of fits. Other interesting markers were alpha-hydroxybutyrate. We saw a significant decrease. So when alpha-hydroxybutyrate is increased, it's proposed to be an early biomarker of insulin resistance, so a decrease would be good. It's also increased in response to oxidative stress, so again, a decrease might be good. Indole propionate is a deamination product of tryptophan by the gut bacteria, and it was increased 2.6-fold, which indicates we're probably having some pretty profound effect on the gut. It's also associated with significantly reduced oxidative stress in neurons, and so maybe overall we have some sort of decrease in oxidative stress. We had a decrease in hypoxanthine, which is purine derivative and a spontaneous deamination product of adenine. So when adenine spontaneously deaminates, you get some mismatch in DNA, and so a decrease in this product would be a good thing. And so these are kind of what and so we also wanted to correlate the change in specific metabolites, pre to post intervention, with a change in the expression of cyclin D1. And so, and Chen Cheng did these analyses, and so I trust them. And so remember, cyclin D1 significantly decreased, and so in general we saw a significant increase in carnitines with the decrease in cyclin D1, except for acetylcarnitine doesn't quite fit. But so here's the p-values and then the different varieties of carnitines, and then the corrected p-value and the R-squared. And again, in nutrition, an R-squared value like this is more cause for celebration because you never see it. And so I think that we can be pretty confident that there is a significant correlation between an increase in carnitines and a decrease in cyclin D1. So carnitines are used to transport long-chain fatty acyl groups into the mitochondria for beta oxidation. And so that fits with our increase in beta oxidation story that we saw from the energy metabolism. Carnitine palmitoyl transferase is a proposed breast cancer target, but I would say that literature is not very strong. Palmitoyl carnitine is shown to stimulate caspases 3, 7, and 8, and that this was our strongest correlation with the decrease in cyclin D1. And the levels of palmitoyl carnitine are shown to increase during apoptosis. And so we also saw a significant increase in glycerylphosphocholine with our decrease in cyclin D1. And we don't have any ones that decided to go the opposite direction this time. They're all significantly correlated the same direction. And so these are the different glycerylphosphocholine, our p-value, and then the corrected p-value. And again, all of these R-squared values are pretty strong. So glycerylphosphocholine are made from phosphatidylcholines, which are the main component of cell membranes. And glycerylphosphocholine eventually entered the TCA cycle. They've also been related to breast tumors, but that literature, again, is controversial. So I think by now we're coming up with a little bit of a story, and I'll see if I can put all of these pathways together for you. And so it seems as though limonene is inducing apoptosis. We saw a decrease in cyclin D1. Cyclin D1 also regulates apoptosis. But I think because we saw a decrease in cyclin D1 in the tumor, but we saw no change in the apoptosis markers in the tumor. And so because limonene is depositing in the fat, it's likely that limonene is just causing apoptosis of adipocytes, or just systemic, non-specific apoptosis. And then our increase in palmitoyl carnitine fits with this induction of apoptosis. And so apoptosis would release phosphatidylcholines, which are metabolized to glycerylphosphocholine, and then we also get cholesterol and other lipids in circulation. We need to induce carnitines in order to metabolize, in order to transport these fats into the mitochondria to undergo beta oxidation. Vitamin C is a cofactor for this process, and we saw an increase in vitamin C. Acetyl-CoA enters the TCA cycle, and beta-hydroxybutyrate is produced as a byproduct, and we saw an increase in beta-hydroxybutyrate. So because we're getting energy from fat, we don't need glucose is shuttled into alternative metabolic pathways, and we see this increase in fructose, amino acids, glycine, alanine, and then the increase in glucuronate and vitamin C. Bile acids are lipid sensing, and so we have a general increase in bile acids. And then maybe the decreased availability of cholesterol to the adrenal gland decreases our circulating adrenal steroids. Those pathways which don't really fit are that increase in indole propionate. This is the tryptophan breakdown product by the gut. The collagen, the increase in collagen breakdown, our decrease in alpha hydroxybutyrate, although if this is an indicator of insulin sensitivity, then maybe this increase in lipid metabolism fits. And then a decrease in hypoxanthine not quite sure where it fits in. But this is what we saw, and it is pretty interesting. And so what have we learned? Well, metabolomics is hypothesis generating. I think we saw a lot of changes that we wouldn't have guessed. In the future, we should target select metabolite classes, so those that changed with the change in cyclin D1, glycerylphosphocholines, and carnitines. Because the ultimate goal for studies like this is to have a circulating biomarker of what's happening in the tissue. And whether or not these are specific to changes in the tissue, I think that's worth further investigation. And I think we've demonstrated that we can use metabolomics as a tool to assess other chemopreventive agents to get a systemic effect. But we still need basic science to go in and really look at those mechanisms, because we've done a lot of correlations but haven't said anything about causality. And so now, I've showed you. All right, we want to characterize metabolic disorders, understand the human microbiome relationship, and response. I just showed you how we can use metabolomics to assess response to an intervention, and inadvertently gave you an idea of why we need to understand the human microbiome relationship because of that one metabolite that significantly changed with the gut bacteria. And then, so now I'm going to show you some of our preliminary data with NAF and why we're kind of excited about it. At least I'm kind of excited about it. So NAF is nipple aspirate fluid, and the epithelial lining of the duct is constantly turning over, and so there's a lot of protein and metabolites in there. And so NAF is non-invasively obtained, and because it's obtained from the breast, it may contain biological information that is unique to, and it can't be found in plasma. There have been some proteomic studies with NAF, and so in one study by Sauter, there were 39 proteins differentially expressed in tumor-bearing breast versus not. And so this is within one individual. And so that shows us that there probably is some different information that you can get from NAF than you could get from plasma. And in a prospective cohort, there are 7 candidate proteins in NAF which predicted breast cancer. There are no NAF versus plasma proteomic profiles or metabolomic profiles, and there are no published metabolomic profiles of NAF. And the reason is NAF is difficult to collect. Not all women produce NAF. In most studies, they've shown that only 40 to 60% of women are able to even produce NAF. But Ed Sauter was able to get NAF from 53 out of 54 healthy women. And so this kind of shows that maybe it's more technique in collecting NAF than it is actual production. Also, women produce a very small sample volume. It's between 1 and 5 microliters. And so when instrumentation wasn't sensitive enough, this small sample volume was crippling and you couldn't get any information from it. But now with advanced technology, this small sample volume is not a problem. And so in collaboration with Imperial, we wanted to just get a general sense of what was in NAF. And we have NAF from 4 premenopausal women, 4 postmenopausal women, and we looked at NAF and plasma from both. It's a very small sample size, but we weren't sure what we were going to find, so we just wanted a preliminary description. And we used NMR and GCMS. And so first we wanted to just characterize what was in there with NMR. And NMR is not very sensitive, and so we had to pool the NAF together in order to have enough sample to see anything. And so I won't read all of these to you, but in general we saw amino acids and sugars. And we saw a pretty high concentration of isopropanol, but we're fairly confident that that's from the alcohol wipe that you had to use before collecting the NAF. And so high levels of glycerol, low levels of other amino acids. And so we have the same sort of data from GCMS. And this is very colorful. So this is a heat map, and it's arranged a little bit backwards, but white is high concentration, dark is low concentration, and so you see a range, they're arranged from low to high, and you can kind of see the distribution of metabolites. Ashley's favorite metabolite, putrescine, is up there in NAF. And so then this is coefficient of variation on different run days and different extraction days. And so it makes sense that those metabolites that are higher in abundance have a lower coefficient of variation. But in general, this is showing the assay is working fairly well, and we can be pretty confident about what we're seeing. And so this is just to give you a sense of what we can see. There's 37 total identified metabolites by GCMS and 23 by NMR, and there's only an overlap of 11. So we can see different metabolites using different instrumentation. And then in the NAF versus plasma, there were 43 identified metabolites from plasma, 37 from NAF, and there are 24 common metabolites between the two. So the next step was, well, are these metabolites which are common between NAF and plasma, do they correlate with each other? Are the levels in NAF similar to the levels in plasma? And so here, this is your metabolite positive or negative correlation, and then the R-squared value. So interesting, the strongest correlation, just lactic acid, is negative, and so they're oppositely related in NAF and plasma. And in general, there's not really a very good correlation at all between what we see in NAF and what we see in plasma. But this is good because it means that there's different information that we can get from NAF than we're already getting from plasma. And so we also wanted to look at the differences between NAF of pre- and postmenopausal women. And sorry, I didn't have the label, but this is NAF, and this is a principal components analysis. So just basically giving you a picture of how different are all these samples from each other. Postmenopausal women are in green, blue is premenopausal women. In general, there's really not a very good separation at all. But Greg at Imperial College is the person that ran these for me, and he noticed my very scientific description of the NAF, which is cloudy or barely cloudy or clear. And he noticed if he took out the clear and not cloudy samples, that we get a better separation, which is interesting. And so he noticed that these that are cloudy and milky, they have more metabolite content than the clear and barely cloudy. And this is interesting because when we looked at the protein content, there was no relationship at all between the way the NAF looked and the amount of protein there. And so there's not a whole lot more we can draw from this, but I think that it's worth further investigation with a bigger sample size. All right, and so then I went to Waters Corporation, and we did the same thing using a UPLC QTOF, and they let me come and play with the instrument. And so same setup, pre and postmenopausal women. They were only willing to run 3 from each group for free, and so that's what we got. So plasma and NAF. And I have a chromatogram for you. And so we were able to detect over 14,000 metabolite features in NAF. And so these are not distinct metabolites. Metabolite features can be isotopes or adducts. But still probably 7,000 distinct metabolites. This is, again, a chromatogram. This is time, only 10-minute run, abundance. And this is the NAF profile, and this is plasma profile. So we can see that there's a lot of very different metabolites contained in NAF than in plasma. And this is all preliminary, so I don't have a lot of specific identifications for you. So Waters is trying to sell you software, and so the output is very colorful. But again, this is just a PCA analysis. This is the NAF, this is the plasma, these are all the labels. And you can see you get pretty good separation between NAF and plasma. Down here, each column is a different NAF sample, and then here starts plasma, so N's NAF, P is plasma. Each line is a metabolite, and so in general, the metabolites which are abundant in NAF are low in plasma, except for this one. And then the metabolites that are low in NAF are more abundant in plasma. And so that's interesting. We also wanted to see the difference between pre and postmenopausal women, and we get pretty good separation. The postmenopausal women all kind of grouped together. The premenopausal women are all over the place. And my guess is this is hormonal, but it's only 3 people, so we can't really draw a whole lot. But it's still interesting that we get such a good separation between pre and postmenopausal women. And then with plasma, again, we get this similar separation. And so we were able to see over 14,000 metabolite features. We were able to identify general classes of metabolites. So there's 2-fold higher levels of steroids, B vitamins, and flavonols in NAF than in plasma, and higher levels of amino acids, phosphorylated sugars, and lysophospholipids in plasma than in NAF. And it seems like there's about 50% overlap in metabolite content between NAF and plasma, but we haven't really gone in and investigate the data enough to know exactly how much overlap. But this is our guess. And so this is just to kind of make a point that we're able to see a lot more stuff with more sensitive instrumentation, like a QTOF rather than GCMS or NMR. And so in conclusion, I've shown you that we can use metabolomics to assess response to limonene intervention. Apoptosis is a likely mechanism, whether it's generalized or related to the cancer. That's warrants a little bit more research. We should look at the glycerophosphocholines and carnitines, and I think we need to always we should start keeping metabolomics in mind when we design clinical trials, controlling for fasting, making sure we get multiple aliquots of plasma, stuff like that. And then I think metabolomics is a really good way to discover new biomarkers and start using other more local biofluids. And so I'd like to thank my mentors, Dr. Chow, Thompson, and Lance. Also Chen Chang for doing the analysis, and everyone in the lab. Heidi collected the NAF, and then the group at Imperial College, Greg did the work with NMR and GCMS, and then Metabolon helped with the metabolite identification for the limonene study, and then Waters did the NAF demo. So are there any questions?
Hold on a sec. We'll get to everyone.
I have a quick two-part question about your limonene study. So you saw a pretty dramatic shift from, or I guess towards beta oxidation. So I wonder, you gave these women 2 grams of limonene. Did you expect that? You said that metabolomics was hypothesis generating, so is there a lot of false positives? And my second part is, were the fold changes biologically relevant, or do you not know?
And so, on to the second part first. Because we just looked at abundance, we weren't able to go in and quantify each one. We have no idea if they're biologically relevant. And that's why I want to go in and, have a standard curve and measure each one to see if, these are really actually relevant. But, and I already forgot the first part.
Are there a lot of false positives?
Yes. But I think because when you're looking at, so there were a lot of metabolites that changed that I didn't show you, and so I picked the ones that were within a class of metabolites. So I think when you see a group of metabolites in one pathway all changing together, that's a little bit more convincing.
Jen, and then back to you.
Sure. So I'm interested in your hypothesis about the increase in the beta oxidative pathway as well. Do you think that you have like an increase in substrate availability to go flux through beta oxidation, or could you, do you still have samples where you could look at actual expression of carnitine palmitoyltransferase? And then my other question is about the beta hydroxybutyrate. I think in Hep G2 cells it will decrease cyclin D1. Do you know if that has an effect in breast cancer cells as well?
I have no idea. And so I do think that the beta oxidation would be increased because of increased availability of lipids. So if we're causing apoptosis, cell membranes are full of lipids, and limonene is depositing in the fat. So if we're apoptosing adipocytes, then there's even more fat availability. And so I think that's kind of why there. But the relationship between beta hydroxybutyrate and cyclin D1, I'm not sure. And also, to answer your question about the expression in mitochondria, we don't have any tissue, and so we can't go in and do those types of studies. We would need liver tissue.
So I think the NAF data is really interesting because it highlights something that sometimes I think we want to ignore, which is that blood isn't a good measure of site-specific changes. But knowing that, is it possible to do your metabolomics studies on like breast tissue lysate or something to get a better idea of what's happening at the site?
Yeah, and so I would say most metabolomic studies in cancer use the actual tissue. And so tissue is harder to get, obviously, but we could potentially do it if we collected it. And so there's pretty standardized methodology for that now, actually.
My question is about the results of the limonene intervention with increase in bile acids. So I was wondering if you guys looked at metabolites involved in cholesterol synthesis after the intervention, and how do you think limonene can possibly affect cholesterol metabolism?
And so I think it's indirectly through apoptosis in the release of extra lipid availability. And so bile acids, that's their job, is to respond to lipids in blood. And so I don't think that limonene is affecting cholesterol metabolism specifically, but indirectly. And then in terms of your other question, looking at other, I've shown you what we saw. And so there were around 300 metabolites identified. We saw, and so I picked out the most interesting ones. And so we definitely have to go back in and do a little bit more pathway-specific investigation.
I had a quick question. I was wondering if you could walk me through what you thought was going on with vitamin C again. I got a little confused there. What do you mean when you're saying vitamin C?
So
Ascorbic acid?
Ascorbic acid.
Okay.
Yeah.
But So you had mentioned that maybe people drank some orange juice that morning and that's why their vitamin C went up. But I got a little confused by the potential pathway that you
So-
also presented.
You can, an alternative glucose pathway is to ascorbic acid through whatever that other metabolite was. And so if you have extra availability of glucose, you can produce your own ascorbic acid. And so it's an alternative glucose pathway. Yeah, I don't know.
Okay, looks like we're done for today. Thank you very much.